Hi, I'm Jen Crainic, and I'm a graduate student in Lucas Sullivan's lab at the Fred Hutch Cancer Center in Seattle. Rather than working on one specific cancer subtype, I prefer to study how cancers from different tissues converge based on similar genetic mutations. My current project focuses on how overexpression of a cell surface transporter, SLC7A11, leads to resistance to certain types of therapies. To study drug resistance, I use cell culture as a model system. We can grow human cancer cells on flat dishes and treat them with different therapies to see how they respond. Look at all of the cells on this dish ready to be used for experiments next week. This particular cell line was derived from a patient with lung cancer. Cells on a dish require media to grow, and it is full of the nutrients that cells would have had access to from the bloodstream. Let's quickly make a bottle of media together to use for our experiments next week. We mix several reagents together to make the most representative fluid possible. We also filter the media to make sure it is sterile so that we don't contaminate our cancer cells with bacteria. During experiments, we add different drugs into the media to test for sensitivity or resistance. At the end of an experiment, we can stain our plates for the total amount of cells left in a well. The darker the well, the more cells that were still alive at the end of the experiment. These cells were the ones resistant to the therapy. We redissolve the state in a clear buffer, and although looking at the plates visually can give us qualitative information about which treatments led to more resistance, we are also lucky to be able to get quantitative information out of these experiments. We can then use our plate reader to measure the absorbance or how pink each well is. This gives us a numerical value of how resistant the cells were to the drug or combination of drugs. We'll use these values to make graphs in order to be able to share our data with other scientists in the field. This project caught my attention because as a scientist, I'm committed to curing cancer faster. I know that although doctors have a large array of potential therapies for each cancer type, many patients don't respond to therapy. Work during the first half of my PhD focused on studying how cells with SLC7A11 grow differently than other types of cancers. The next question I wanted to ask was how these cells grow differently in response to therapy. In this recent work, we have found a new mechanism of drug resistance tied directly to overexpression of SLC7A11. I'm excited about this new discovery to be shared with scientists and physicians in order to better inform patient treatments.